Thank you for joining us for World Migratory Bird Day Live. We want to take a moment to honor and express our gratitude to our World Migratory Bird Day supporters. Your commitment and support are crucial to the success of migratory bird conservation. A special thank you to our global flyway partners, the U.S. Forest Service and National Park Service. We are truly grateful to our American flyway partners, the U.S. Fish and Wildlife Service, Bureau of Land Management, Optics for the Tropics, and Partners in Flight. Your dedication to conservation and education in protecting migratory birds is invaluable. Thank you for your contributions. Thank you to our program partners, the Xerxes Society for Invertebrate Conservation, Pepco Holdings, Avian Powerline, Songbird Essentials, the U.S. Department of Defense, and Vortex Optics for their partnership and support. To our friend-level sponsors, Birds Caribbean, Nature Canada, Birds Canada, and Canabio, your support is appreciated in achieving our goals of fostering community and conservation across borders. Lastly, Environment for the Americas is grateful to all of our members, donors, supporters, and World Migratory Bird Day event organizers. Your contributions are helping to create a more connected and conscientious world. And to you, our audience, thank you for watching and supporting migratory bird conservation. You can further support our campaign by visiting migratorybirdday.org or by donating to Environment for the Americas at environmentamericas.org. Thank you for watching World Migratory Bird Day Live.
Broad-tailed hummingbird, and they probably do some pollinating of this species, although it's also visited quite heavily by the queen bumblebees, a good pollinator of that species. So we know from our work on flowering phenology that the floral resources are changing, and they're changing in different ways. And not all the wildflowers are changing their timing of flowering at the same rate. And their changes in abundance from year to year are also not occurring at the same in the same directions. So those floral resources are changing differently for different pollinator species, depending on which of the wildflowers they happen to be visiting. In addition to the wildflowers, another potentially important resource for the hummingbirds is stream insects, particularly for the nesting females while they're feeding their fledglings. And if you look carefully in the middle of this picture, you'll see that there is a hummingbird nest right next to the East River, just outside Gothic, outside the Rocky Mountain Biological Lab. There's the female sitting on her nest. The eggs hatched successfully, but unfortunately a crow found that nest and apparently ate the nestlings. So as you all know, the climate is changing. Here's a picture that shows how the temperatures have changed dramatically over the time that temperatures have been measured across the country. And note that where I work down here in Southwest Colorado is one of the hotter hotspots across the continental United States. And so that's those changing temperatures are certainly going to have an effect on both the wildflowers and potentially on the pollinators. I've been doing a lot of analysis this winter of the Crested Butte weather station data. Those data date back to 1910. And one of the things that I've looked at is how many rainy days have there been during the summer from May to September. And note that this is a curvilinear relationship. If you look at the whole 110, 114 years of 113 years of data for this metric, particularly over the last 30 or so years, there's been a very strong decline in the number of rainy days during the summer. The mean number of consecutive days with no rain has been undergoing a curvilinear change here, but seems to be moving towards a regime of having a higher number of consecutive days with no rain during the summer. One of the predictions of many models of climate change is that the climate variables are going to be becoming more variable during the future. And so statistically, we can look at this by a measurement called the coefficient of variation that tells us how variable is the metric that we're looking at. And so I calculated that for the amount of rain per rainy day in Crested Butte. And you can see that over that time span since 1910, that metric has been getting more variable. Up in Gothic, we have a shorter time record. It dates back to about 1975, courtesy of Billy Barr. And one of the things that he's measured is, when does the snow melt? And you can see it's been highly variable. It's been as late as the 19th of June. And that was a winter that I happened to be spending the winter up in Gothic. And it's been as early as the 23rd of April. So lots of variation, but there's a trend. It's not yet statistically significant, but a trend towards earlier snowmelt at 9,500 feet. I've been able to reconstruct what was some of the history of snowfall and snowmelt in Gothic by looking at a stream gauge down in Almont, Colorado, which is on the East River, which is the river that flows through Gothic. And because there's a very strong correlation between how much snow falls in Gothic and what happens to the stream down in Almont, I can reconstruct how much snow did we get back to about 1934 and when did it melt. And you can see that it hasn't changed too dramatically over that longer time period, but there is overall this trend now towards lesser snowfall. So let me talk a little bit about some of the specific research projects on hummingbirds that have gone on at RMBL. So here's a feeder outside my cabin at the lab. You can see this is a banded male broad-tailed hummingbird, and it also has a pit tag glued on its back. So this is the kind of tag that a veterinarian might inject under the skin of your cat or your dog, so that if it gets lost, that somebody else can scan the animal and get the code off that tag and identify your lost pet. Well, we can do a similar thing with hummingbirds. This was a picture taken several years ago, and we're actually now using tags that are about half the size of this one. They're glued onto the back of the hummingbird, and as the hummingbird molts its feathers, that tag will lose its grip and it will fall off. So they're only carrying it around probably for a month or two at most. I've been doing some testing for a company called Bird Buddy that makes bird feeders with cameras built in, and they have a prototype hummingbird feeder now. And I tested that for them last summer, and that camera took some images of the hummingbirds visiting. So for instance, here are two hummingbirds at my cabin, that's Crested Butte Mountain in the background, and they use artificial intelligence to try and identify the birds. And you can see that this is a somewhat questionable identification, saying that this was a visit by a house sparrow to my hummingbird feeder. So one of the things that they're working on with their beta testers is trying to train the artificial intelligence model that will come up with more realistic identifications of the hummingbirds. A graduate student who finished his research a summer or two summers ago now, I guess, at the Rocky Mount Biological Lab is Nicholas Alexander. He was a grad student at Berkeley working with Dr. Noah Whiteman, and his PhD work examined the genomic coupling of two different aspects of hummingbird ecology. One is their bill size, including bill length, and then also their migratory arrival dates, their phenology. So here's a graph showing a plot of the day of the year of the first capture of different individual birds. So some of them tend to arrive early, some of them tend to arrive later. There's a bit of a drop there in the middle. And in terms of the morphological measurements, he would measure on each bird that he caught the exposed culmen, the full gape, getting those two different measures of the hummingbird bill. And you can see that they're different for males and females, that the females' bills are a little bit longer, and also in both those measures, they're a little bit longer than those of males, which are the gray bars. And in terms of the timing of the season, birds that arrive early in the season tend to have somewhat shorter bills as measured by culmen length or full gape than the birds that arrive a little bit later. So it's interesting that there's that correlation between phenology of arrival and the size of the bills. He also measured, although we're not reporting the results here, the differences in bill curvature and the bill width. So he took lots of measurements on those birds that we captured. This is a plot showing the different species that these hummingbirds use, including the early bluebell, the glacier lily, the larkspur, willows, columbines, Indian paintbrush, a twinberry, all the way on up to a thistle flowering later on in the summer. And if you plot the corolla length, the size of those flowers, against when they come into bloom, you can see that there's a trend towards flowers blooming with longer flower tubes later in the summer. He also measured or collected samples of the pollen that these birds have on their bills. So you can see that this female broad-tailed hummingbird has quite a bit of obvious pollen on her bill. And by using a piece of sticky tape, he could get a sample of that pollen, put it on a microscope slide, take it back to the lab and try and identify it. And this graph shows how does the size of the bill measured by the exposed culmen length match up with the pollen that he found on the bill. So Ipomopsis, the scarlet gilia flower, and the birds with shorter bills tend not to have much pollen from Ipomopsis on their bills, whereas birds with the longer bills have quite a bit of that Ipomopsis pollen. The opposite was found for the Mertensia, the bluebell pollen, tended to be more common on birds with shorter bills. So those morphological traits are helping to determine which flowers the hummingbirds are visiting. He also was able to identify the genotypes of 500 of those broad-tailed hummingbird individuals that we caught in Gothic. So he knows every gene in those 500 individual hummingbirds. And this is a picture showing what are some of the genes. These are names of individual genes that he identified in the hummingbirds, and some of the genes that were found in studies of other species of birds that other people have done, of genes that are influencing apparently bill size in other species. And then there are a set of interactor genes as well. And he also was able to identify a couple of genes that apparently influence are linked to the arrival time, whether it's birds that arrive early or birds that arrive late. And there's some work from previous studies that also show that. So he's been able to identify now at the level of individual genes of individual hummingbirds, what are the genes that influence the size of the bill of those birds, as well as when they arrive at the breeding grounds. Something else he was able to calculate was an inbreeding coefficient. And maybe you know enough about genetics to know that inbreeding is considered not to be very good, because if you're breeding with a close relative, you're more likely to end up with genetic problems in your offspring. Hence the genetic problems that royalty in England used to have when they were marrying close relatives like first cousins. So fortunately, it looks like the inbreeding coefficient is not very high for hummingbirds. So they're not likely to be experiencing those kinds of genetic problems. Another major research project on hummingbirds at RNBL has to do with their vision. And I want to define something called non-spectral color first. So a non-spectral color is a color that's not present in the visible spectrum, what our visible spectrum is, and can't be produced by a single wavelength or narrow band of wavelengths of light. And so these are colors that are evoked, whereas colors that are evoked by a single wavelength of light are often described as being produced by monochromatic light. And we do see some colors with our vision of non-spectral colors, and those are examples include magenta, pink, cyan, and brown. So blue and red together create that magenta color that our brain tells us is magenta. Red and purple together create what's called pink in our minds. blue and green produce cyan, red, yellow, and blue produce brown. So we do see some of these non-spectral colors. Hummingbird vision is different because instead of seeing 3 primary colors as we do, they can see 4. And one of those is ultraviolet light, which we can't see. So I'm going to show you a graph or two from a paper that was organized by Cassie Stoddard, who's a faculty member at Princeton University, who got interested in hummingbird vision and started coming out to the Rocky Mountain Biological Lab maybe, I think maybe 6 or so years ago now, and asked if I would help her with this project because I happen to have the permits that are needed from state and federal agencies to work with, to catch our hummingbirds and to band them. And so we were able to look at what are the predicted wavelengths that hummingbirds are likely to see, including iridescent colors on this broad-tailed male. And therefore, what are some of the non-spectral colors that hummingbirds, in theory, should be able to see? And here's a plot of the different colors that they can see, and these intermediate colors, that would be the non-spectral colors. So she did a set of experiments on some of the primary visual pigments that they see, as well as this whole long list of non-spectral colors that hummingbirds in theory could see. And the way she did this was to put out 2 feeders. Each feeder had a light source associated with it, and she could change the light source with each feeder. One of the feeders had sugar water, the other feeder had plain water. And she wanted to see, could the birds use those visual cues of what color the lamp was putting out to tell them which was the rewarding feeder. And the probabilities down here at the bottom are the probability that they chose the right feeder. And if you have 2 of the same colors, there's a pretty small chance that they can tell which one. There's about a 50-50 chance that they'll pick either of those feeders. Whereas with a color that they can discriminate, for instance, UV plus red in the light versus just red, they can tell pretty quickly which one has the right reward, has the reward in it, and almost 100% of the time they can pick the right feeder. So that was able to give a good clue through these experiments which feeders they can visit. And I mentioned those pit tags that we put on the hummingbirds. And so she had individual birds that would come to the feeder. Each feeder also had an antenna on it, so she could identify the individual birds as they came into those feeders through those pit tags. So Cassie and her co-principal investigator on a National Science Foundation grant have been looking at behavior. What colors can they discriminate through their vision? Looking at their foraging, what flowers and what colors do they visit? Looking at the morphology of their eyes to look at how do their cones and rods work to see those different colors. Looking at their neurology of their vision, look how do their retinas transmit information to the brain and what parts of the brain are involved. And then also the genetics. Are there differences among hummingbird species or among individuals in their vision? And the last hummingbird research project I'll mention is one that has its origins back in the 1970s when Bill Calder realized that male hummingbirds, male broad-tailed hummingbirds, seem to fly up in altitude at night or just to spend the night. So he noticed that as the birds in the evening are taking their last visits to the feeders, that he would hear them flying up in altitude. And he tried going up in altitude to try and hear how high were they going and try and figure out where they're spending the night. He wasn't able to figure that out. But about 6 years ago, some companies started to make radios that were small enough to put radios on hummingbirds. And I was able to get hold of 5 of those, paid for with some grant money, and to try putting those radios on male broad-tailed hummingbirds to track them. And it was marginally successful. I had trouble with keeping the radios on the birds. I had some birds that I put radios on that apparently were transients, and I never heard them again after I abandoned them and put the radios on. But I have one data point where I heard a radio transmitting from this spot, which was 10,624 feet. And I had put the radio on this bird down at my cabin here in the lower right part of that picture. And my guess is that when I went up, I was actually able to find the radio at the base of a tree, that the bird had managed to take the radio off, or the radio had fallen off. Maybe it wasn't glued on well enough, but I'm pretty sure that's where the bird spent the night. And so I wanted to find out, well, why would a bird fly over 1,000 feet up the mountainside to spend the night? And one possibility was that it's a temperature difference. So I put a transect of temperature loggers from down at the East River up to, and then a little bit higher than where I found that radio. And then I measured how cold does it get during 4 days at the beginning of July back in 2017. And you can see it was below freezing down by the river at night, whereas it was some 20, what is that difference between about 28 degrees, 10, 14, about 16, 17 degrees warmer at night at that higher altitude where the bird was spending the night. And so my guess is, and we haven't finished making the calculations yet, is that it's worth the bird's while energetically to fly more than 1,000 feet up in altitude to spend the night where it's so much warmer, and then fly back down the next morning. The females, as I showed you earlier, seem to be tied to the bottom of the valley, much lower elevation, just to make their nests. And that may be in part because they're so close then to the streams where they can catch stream insects to feed their nestlings. And the streams may also keep it a little bit warmer at night. And by also building their nests in trees where there's one branch below a branch just above their nest, that helps shield them from the cold night sky. So they're probably picking microhabitats that are as warm as they can get while they're still staying close to those food resources at night. I'll finish up with this graph showing the day of the year that Billy Barr has first seen robins up at the Rocky Mountain Biological Lab. And over the years, the robins are arriving earlier and earlier, to the point where in one winter, I think this was 1995, when I spent the winter out in Gothic, that we looked out our window one morning, and this was probably in early May. There's still probably almost 5 feet of snow on the ground, but the first robin was showing up. Not likely to find any worms there. So in conclusion, we know that migratory hummingbirds are changing their phenology of migration. The wildflowers that they depend on are also changing, but not in synchrony with either the migration or with each other. Flowers are doing, different species are doing different things. The floral resource, the wildflower phenology and abundance, are dependent on the environmental cues like snowpack and snowmelt dates. This climate change is likely to produce earlier and less abundant flowering of hummingbird flowers. And that change, or this change that's ongoing between plants and pollinators, is going to produce, presumably, changes in the evolution of both of those partners. And these genetic insights, genetic studies of hummingbirds, are providing a variety of new insights into their ecology. The hummingbird vision is the current focus of research, and banding that we're doing in their recaptures has the potential to help monitor these changes. It's also told us that these birds are very philopatric. They tend to come back to where they were born. So the oldest known hummingbird in the wild is a female that, I forget whether Bill Calder or I first banded it, but we saw that bird for almost 13 years, or almost 12 years, coming back to Gothic. And so she was at least 13 years old, because she was at least one when we banded her. And so that's the oldest known hummingbird in the wild. I've caught other birds that I know are 9 years old, coming back to the same place year after year. So I hope you learned something about hummingbirds. If you're in Colorado, I'll point out that the Mountain Film Festival in Telluride, which is later this month, is actually going to have a short film, an 11-minute documentary that was made by this young filmmaker, Brendan Young, um, and about the work that we're doing with hummingbirds and the fact that my son and my granddaughter have also been involved in this research that is ongoing. So um, that'll be the world premiere of this little documentary about some of our work, including the hummingbird work. Thank you for your attention, and I'd be happy to try and answer any questions that you might have.
Thank you so much, David. That was, as always, amazing and so great to learn about your research and what you're working on. I wanted to take you up on something we talked about before the presentation and see if you had mentioned that you had done some insect surveys there, and I wanted to see if you had any information about hummingbirds and insects and their dependence on insects. Partly because this World Migratory Bird Day, we're also focusing on insects and how they support bird populations.
In the context of hummingbirds in particular, we already know that they're eating a lot of stream insects. In fact, it's a stream biologist at the Rocky Mountain Biological Lab who found that nest that I showed a picture of, because walking up and down the streams collecting his insect samples, he sometimes frightens birds off their nests or sees them going to land in a nest. So I was able to take a bird that had died as a window strike and give that to him. He dissected some of the fat out of that bird and is going to do a chemical analysis that would tell him how much of the fat on that female hummingbird comes from stream insects, because the aquatic insects make a particular kind of fatty acid that is not made by terrestrial insects. So we may know, after he completes that analysis, how strong is the link between nesting hummingbirds and aquatic insects. The other aspect that I can comment on is a project that was begun by Dr. Michael Soule, who's also often credited as being the father of the field of conservation biology. He used to work at the Rocky Mountain Biological Lab back in the 1980s and started a project putting out an insect trap called a malaise trap. And every week he would put the trap out for 2 days a week, all summer long, and collect any of the insects that got caught in it. And he continued that for, I think, 5 or 6 years. And then when he was going to stop his work at the Rocky Mountain Biological Lab, he asked if I'd be interested in continuing that project, which I have. And so now we have a decades-long record of the insects that are caught in malaise traps. Now, not all of those are eaten by hummingbirds. Some of them are too big for hummingbirds, but probably a lot of them are eaten by insectivorous birds. And as probably a lot of this audience knows, the decline of birds worldwide, or certainly here in North America, has been particularly severe for insectivorous birds. So one possibility is that the reason they're declining is because a loss of their diet. And that may be because in part of climate change, it may be in part because of the increased use of insecticides or pesticides or herbicides, things like neonicotinoids that are now on almost every seed, corn seed, wheat seed, and soybean seed that are planted in the United States, for instance. They're pre-coated with these neonicotinoid pesticides before they're planted. So we thought that, well, our research site, there's no agriculture. There's been no change in land use. There are no new subdivisions. There are no street lights at night. And so some of the factors that have been hypothesized to be responsible for the strong insect declines that have been recorded in some other long-term studies around the world, particularly in Europe, those aren't problems here at the Rocky Mountain Biological Lab. But we were shocked when we did the close analysis and published a paper last year to find that there's been a 60% decline over the years that we've been monitoring insects here at the Rocky Mountain Biological Lab. So that potentially could be a strong impact on both hummingbirds and other insectivorous birds. We don't know for sure why there's been that strong decline. It may be related to the warmer summers, to drier summers, but it also may be related to things like the the chemicals that are being spread around the world through rain and snow, some of which are endocrine disrupting chemicals that influence the physiology and metabolism and the endocrinology of both vertebrates like ourselves and invertebrates. So that's still an open question, but what's not in question is the strong decline.
Yeah, thanks. I would take that one step farther. You talked about the phenology of the glacier lily and other flowering plants that hummingbirds depend on. In your work on insects, have you also noticed any phenological shifts or shifts in timing of emergences or any other activity on insects that could also impact then, insect-eating birds like hummingbirds?
Yeah, the phenology of almost everything up there, from wildflowers to marmots to hummingbirds um, to bumblebees, their seasonal phenology is pretty much determined by when the snow melts up there. Because it's not until the snow melts that the ground starts to warm up and the bumblebees get the signal they should come out from underground and start their nests, their colonies for the summer. Um, so it's probably a lot of this the same phenological, the same climate variables that are strongly influencing the phenology of just about everything up there. 15 years ago, Dr. Rebecca Irwin started a project. She's a faculty member at North Carolina State University, was already working at RMBL, and I asked her if she would be interested in collaborating with me on this National Science Foundation funded project on phenology. So she added a bee component. So every couple of weeks, she goes to the same meadows where we're measuring the wildflowers and sets out pan traps and uses butterfly nets to catch the bees. And so she's got a 15-year data set now for the bee phenology and abundance. And we can begin to put those two together and see how closely linked are the insects and the wildflowers. We don't have a lot of answers to that yet because she's still working on those data and we're still collecting those data. But we do know, for instance, that bumblebees are responding to the changing climate by moving up in altitude. There are some low altitude species that we now see in Gothic that we didn't used to see there. And there are probably a variety of other insects for which we haven't been keeping close track that are also moving up in altitude. There's at least one mosquito that used to be common around Gunnison, but is now present up at Gothic, and it's a carrier of West Nile virus, and so could potentially be a problem for the birds up at higher altitude, whereas that mosquito wasn't present in the past.
Mhm. Yeah, so there isn't a whole lot of data on, of course, hummingbird diets. Do you have any information on about how much of a hummingbird's diet is insects?
Well, we do know that they're getting almost all their protein from catching insects. There's a little bit of protein in the form of amino acids in the nectar that the plants they're visiting, but that's not a huge amount and probably not enough to keep a hummingbird going or build a baby hummingbird. So I'm guessing that a pretty substantial amount of their protein is coming from the insects that they're catching. I'm collaborating with another researcher, Dr. Rankin from UC Riverside this summer. I'm going to be collecting fecal samples from some of these hummingbirds by putting them in cages with a piece of filter paper in the bottom of the cage and waiting for them to defecate and then sending her those samples. So we may have a better answer to your question after this summer's field work.
Okay, great. We'll bring you back in the fall and we'll hear the results of that. I see it. I'll do one more question from the chat here. I guess the question was about recommending pollinator studies in local communities that folks could participate in. What would be some helpful first steps to launching, say, a local or a smaller pollinator research project that could help inform us for the benefit of migratory birds?
Well, there are a variety of citizen science projects related to plants and pollinators. One example is the USA National Phenology Network, and you can find that by doing a Google search, or I think their website is usanpn.org. And they rely on citizen scientists collecting data on when plants in their backyards come into bloom, So anybody can collect those data anywhere and record them on the USANPN website for scientists to have access to those data. There's a similar project for bumblebees, looking at the phenology and the abundance of bumblebees. In fact, I just got an email message today from a Western bumblebee watch project that will help train people to identify bumblebees and has this protocol for how to catch, photograph, and then report those bumblebee sightings. In terms of hummingbirds, things like eBird or iNaturalist, those are important ways for people to record their observations of birds or the flowers that they're visiting and make those data available to scientists. The Xerces Society was mentioned earlier, and they also have a number of projects where citizens can help by planting pollinator gardens. There are recommendations about what plants to plant in your backyard to attract pollinators. The USA National, the National NAPSI, North American Pollinator Protection Campaign, is another group that provides information about what plants to put in your yards, regional specific recommendations in order to promote pollinators. So there are lots of resources.
There are a lot of resources. I'm going to give one more shout out to Journey North, which is a simple, also citizen science program where you can record first arrivals of hummingbirds as well as nectaring. So recording the flowers that they use. I'm going to give you one more question. I think maybe I have 2 more minutes here. From
Okay.
Todd Eskelin from Alaska, who asks, with a relatively short generational time, would you expect that genetically controlled influences like bill size have a fairly quick impact on the morphometrics of hummingbirds through natural selection based on changing floral availability or insects that could be captured more easily because of bill shape and size? Maybe you were going to say there. Yeah.
An interesting question. And yes, I think based on other studies, we know that that kind of evolutionary change can happen quite rapidly. The example I can point to is Darwin's finches in the Galapagos. And because the grants research on those birds, long-term research, they were able to show an effect of things like a severe drought, which affected the size distribution of the seeds that were available for those Galapagos finches to visit. And there was a response in the average bill size of those Galapagos finches over the period of only several years. So probably those changes can happen relatively quickly if there's strong selective pressure.
Wonderful. Thank you, David. As always, a fascinating presentation that leaves us with so many more questions, but having learned so much too. So thank you for joining us today. And thanks to everybody who is also joining us for Migratory Bird Day. This is the official week. And I hope that you're able to join a World Migratory Bird Day activity or event near you or more of our presentations this week. So thank you again, David. Thank you so much.
You're welcome.
And Miguel, I don't know if you're going to come back and close us out.
Y bueno, muchas gracias también al doctor David por la presentación. Solo para concluir, en español, nos vemos el día de mañana para poder continuar con la programación del Bird Day Live con actividades de América del Sur y el Caribe. Entonces vamos a invitarlos para el día de mañana a continuar celebrando el Día Mundial de las Aves Migratorias y aprendiendo a proteger los insectos y las aves. Muchas gracias. Thank you very much, Dr. David, and thank you, Susan. We are now just complete the session.
Thank you for joining us for World Migratory Bird Day Live. We want to take a moment to honor and express our gratitude to our World Migratory Bird Day supporters. Your commitment and support are crucial to the success of migratory bird conservation. A special thank you to our global flyway partners, the U.S. Forest Service and National Park Service. We are truly grateful to our American flyway partners, the U.S. Fish and Wildlife Service, Bureau of Land Management, Optics for the Tropics, and Partners in Flight. Your dedication to conservation and education in protecting migratory birds is invaluable. Thank you for your contributions. Thank you to our program partners, the Xerces Society for Invertebrate Conservation, Pepco Holdings, Avian Powerline, Songbird Essentials, the U.S. Department of Defense, and Vortex Optics for their partnership and support. To our friend-level sponsors, Birds Caribbean, Nature Canada, Birds Canada, and Canabio, your support is appreciated in achieving our goals of fostering community and conservation across borders. Lastly, Environment for the Americas is grateful to all of our members, donors, supporters, and World Migratory Bird Day event organizers. Your contributions are helping to create a more connected and conscientious world. And to you, our audience, thank you for watching and supporting migratory bird conservation. You can further support our campaign by visiting migratorybirdday.org or by donating to Environment for the Americas at environmentamericas.org. Thank you for watching World Migratory Bird Day Live.